I was treating AML in 1977 with two drugs that we refer to as 7 and 3. Today in 2019, I'm doing the same exact thing. And I really don't see much changing. What would be your suggestion to do things differently so things change by in the next 10 years by 2029?
Well, I think one of the issues that we've had in cancer therapy is that since Ehrlich, since the magic bullet hypothesis developed, there's been an enormous effort to find magic bullet drugs, and so the addition of drugs, the search for drugs, has now been, has resulted in a vast industry and extensive profits for people. But and it would be great if we could find magic bullets, but I'm not sure that's feasible. And what we've not done, I think, is to consider how we give drugs now and whether we're giving drugs in a way that makes the most sense, and how we sequence drugs one from another. They're often different manufacturers, and so there's been really no support for this idea that maybe if we have treatment one, the treatment two should build on treatment one, and so forth, so that we're then applying a kind of strategic sequence of drugs rather than just what's whack-a-mole kind of thing where you have one drug, you use it until the progression, then you use another drug until progression. I think that's I don't think that's an optimal way to do it. So one of the things that I've come up with, I think we already discussed in our email, is this idea that once you've achieved let's say an NED, or let's say a significant response with a drug that you have, giving the same drugs is not useful. You've selected resistance. It's futile to continue that process. On the other hand, you've knocked the tumor down quite a bit, and it's in a state that's quite vulnerable, but you can't keep using the same drug to do that. You have to pull away the drug, and I know that that's something that the oncologists don't find conceptually appealing. To take a drug that's been very effective, you keep using it, and I think that's a fundamental error. I think that you have to stop using it. It's been great. You've done the best you can do. Take it away, and now apply another one. Use our understanding of evolutionary dynamics to do this. And if you can control the tumor, if you don't think you can cure the tumor, then use the evolutionary dynamics to at least control it for as long as possible. Or if you think you can cure it, use the evolutionary dynamics of extinction to develop strategies. But what we do now, and it's almost dogma as far as I know, maximum tolerated dose continuously until progression. By the time the tumor is growing again, it's too big. Your second line therapy is not going to be very effective. But you had it on the ground, and we talked about this a little bit, that it's like, a boxing match where, the rules are, and the oncologists are very gentlemanly about this. And I'm sorry, that was a sexist comment, but I don't know what ladylike maybe is the other, is that, if you knock your opponent down in a boxing match, you have to go to the corner and stand there until the opponent gets up and, is better. And that's almost what we do with the cancer. We wait until it gets back up. Whereas this is a knife fight. This is, when your opponent goes down, you just keep kicking and hitting. And I mean, that's the goal. And I don't think that we've been using that analogy as well as we could. So anyway, that's a long answer. But I actually think that we keep looking for more drugs. We keep thinking, okay, next week, next year, next month, a new drug will be developed, and then we'll add it to what we have. Whereas I think we actually may have enough drugs, but we're not putting them together in a way that conforms to any conceptual model. We don't have a model that we just keep blasting away at it. And I think that if we organized ourselves, organized the therapy better, that we would have better outcomes.
Yes, this is what intrigued me after reading your paper, that your basic assumption is that we may already have effective drugs. We need to use them more intelligently, taking into account the evolutionary perspectives of cancer proliferation, which makes total sense to me.
Yes, by applying maximum tolerated dose until progression, we're doing a probably what's evolutionarily the worst way you could do it, and it's called selective release. So what's happening is that you're killing a lot of the population, but you've got resistant cells there, and by eliminating all their competitors, now they're free to proliferate, and now they compete against each other. So they're not competing with the sensitive cells, they're competing with each other. And because of that, there's going to be this progressive increase in fitness. So the cost of resistance, will be compensated as they compete with each other. They'll develop ways to get around that, and so they'll begin to proliferate faster. That's a bad place to be. So if we could have them just compete against each other, compete against the sensitive cells, and then they're competing against another species essentially, and so they can't select intraspecies for their own fitness because they're fighting with each other. But if you eliminate all their competitors, you're basically opening the field for them to develop as quickly as possible, and in the process they're competing against each other, and now they're going to evolve ways to proliferate faster and still be resistant. And so that's, to me, is a fundamental error. This, by the way, is not this is not rocket science. Farm people who run farms have been doing this for 50, 60 years. This is not, we're well behind in terms of our understanding of this. This is not really cutting edge evolution. So.
I think it makes perfect sense to me. Thank you so much. My next question is, there are over 3.5 million papers in cancer. 137,000, I believe, just in 2018. But there is a staggering disconnect between our great scientific insights and our ability to translate that into improved therapies.
Yes.
What are we doing wrong?
And I'm sorry, I'm going to go back a little bit. I'm a radiologist. I actually had a physics background in college, then went to medical school. And then it happens that my first job was at the Moffitt Cancer Center in Philadelphia. And I really didn't know much about cancer at the time, but I wanted to learn more because I wanted to try to make a contribution. And one of the things that struck me is that individual papers are really good. So if you read through the Cancer Research Journal, for example, each paper by itself seems really good and really important. And then you read another one and that seems really good and really important, but you can't figure out how they connect with each other. So in physics, they develop a conceptual model. There's a framework of fundamental principles that everything rests on, and that framework is lacking in cancer biology, in my opinion. And so the way physicists do this is with mathematics. So the theories are always framed mathematically. And so, I started doing it. To say that people hated it would at Fox Chase would not do justice to how negative they were. But here, we have 9 mathematicians at Moffitt now. We have 2 evolutionary biologists, all on the faculty, with the idea that we would provide a theoretical framework to help people understand and to fit all these different pieces, the literature into a coherent whole, rather than letting them just kind of go. And it also can guide people. One of the things that I think happens is that people get, cell cultures, they do some perturbation, they get some result, and then they publish it. It's not clear that has anything to do with anything. I mean, because the cells in culture have evolved to be culture phenotypes. Whether that has anything to do with an in vivo, much as a clinical phenotype, is not really clear. Then we've kind of lost that. In my opinion, the other thing is that we've gone genetic, and I think in part that's because it's relatively easy to use this fabulous technology to get results and publish the results, but it's frequently not clear what that means. So I had a conversation with one of our physicians here who noted that there's a mutation that was associated with resistance to chemotherapy in lung cancer. And so the question was, well, what does it do? And it was, well, it confers resistance. But that's not, the question is, how does it do it? What is the mechanism by which resistance occurs? You need to understand that, but increasingly we think if you know the gene, you know everything you need to know about something. And I think that's a problem. And I think that even when we talk about evolution, cancer biologists talk about evolution as though it was a genetic process, whereas Darwin viewed evolution as an interaction between environmental selection forces and phenotypic adaptations. It's useful to think whether Darwin could have, if he was a modern biologist and had molecular biology equipment in the Beagle, if they brought him just random samples of finches from the Galapagos Islands, could he have written on the origin of species? And I suspect the answer is no, because what he saw was the beak of the finch and the seed had morphologic similarities. That it made sense. You could understand it because there's a cause and effect, whereas the genetics are often one step back from that. And so sometimes by thinking that we understand a cancer because we know the mutations that are associated with it, I think is probably missing the point.
Yes. Excellent answer, as expected. My third question is that the fact that children respond to the same treatment better than adults seems to suggest that the biology in childhood cancer is different. But of course, it could also mean that the host is different and that we have good therapies for even adults, but because our bodies are so decrepit by the time we are getting cancer, that no matter what we do, it's not going to work. Having effective drugs may not be enough. What is your solution for that?
Well, I've started to work with pediatric oncologists just recently. Moffitt is an adult hospital, but we now have a young adult program, and that's particularly for pediatric sarcoma. And so I learned a lot about this. I honestly have never worked with childhood cancers before. And so if you look at childhood leukemia and how it's treated the same way that you see Anthropocene extinctions. So they start with a therapy, and they've done this by trial and error. So they start with a therapy. It's very effective. Sometimes you don't see any cancer cells in the marrow at all, and yet historically they know that it comes back.
Yeah.
And so, they've now instead of just keeping the therapy going, which is kind of standard, they stop it and they give another therapy. They give this acute consolidation phase, and then they give a moderate consolidation phase, and then they give a longer control kind of phase. But what's I think important is that they realize that even though they can't see the cancer, it's still there, and they recognize that continuing to treat it with the same drug is futile. And so what they did was they treat the drug before they treat the cancer before they can see it. They know it's there. So if they waited to see it, it would be too late. So they immediately began therapy, and it's a different therapy. and then they give a different therapy and a different therapy. So I think the advantage is that they've got a lot of drugs for leukemia. And the thing is that if you're treating a population that's been badly damaged, so you've knocked it down, there's populations that are left, but they're survivors and they're fragmented. They're probably relatively homogeneous, and you may not need a drug that is as powerful as one that would ordinarily take a big tumor and knock it down. Your first phase drug needs to be pretty powerful because you need to bring the population down. The second and third and fourth phase drug may just need to kind of to just hit it a little bit, because in this setting you can get what's called demographic stochasticity, subtle variations in birth and death rates. So a slight decrease in birth rate or a slight increase in death rate can cause a small population to go extinct very quickly. So you don't need to blast it with everything you've got. Now you can use more subtle effects. And so even a drug that's not terribly effective against the big tumor may actually be extremely effective in that setting. And so I think that one of the things that, to me, that would be important is to start looking for drugs that, now we require that every drug have a monotherapy, be effective against, in monotherapy. But a drug that gets a 20% reduction in the cancer in a big tumor may not be approved for that, but a 20% reduction when you've got a small population, it may be sufficient to drive it to extinction. So I think there's two different phases of treatment, and I think that because we've only looked at drugs that will treat the big primary tumors or a big tumor burden, that we've neglected opportunities to find drugs that, when you've knocked the tumor down, could actually be very effective in the kind of delivering it the death blow to it to cause it to go extinct.
Mm-hmm. Interesting. Well, you have great knowledge and experience in the field, Bob, and if you were given limitless resources, then money is not a problem, what would be your solution to finding a cure for cancer?
I think we have the cure for cancer. I think for many cancers. My goal would be to convince the adult oncologists to start using a different approach. Let's find a drug. If we don't have it, you've got to have one effective drug that will knock the tumor so that it gets to an NED or at least a very small tumor burden. And we have sort of a baseball analogy. You need a starter, and that starter has to go for 7-8 innings. If you don't have that, you at least need a middle reliever, something that will come in. So you've got the best result you're going to do. Now bring another one in, and then you need a closer. You need somebody. You need a drug that's going to take a tumor that's beaten, that's on the ground, and finish it off. And I think we can do that. I think we could do that in metastatic prostate cancer. I think, there's certain areas where you can get a very good response to the initial therapy and then it comes back. I think we could use that to do better. I think, so we have good math models for this now. We're starting to do some preclinical experiments, and I would probably do more of those. But I think that the goal of preclinical experiments should be to test the basic principle and not necessarily specifics. I think the specifics are going to have to be tumor specific. And certainly if you have pancreatic cancer, there's no good drugs for that really. So that's not a curative sense. So let's start thinking about how, what are ways that you could do it? Because you have radiation therapy, you could try, immunotherapy. There's a number of things that may not be terribly effective when you've got the big primary tumor, but may be extremely effective if you can get it, if you can knock it down. So I don't plan to have unlimited funds anytime soon, but yes, that's what I think too. But I think we're closer than we think we are.
Great. My last question is a little bit philosophical. And that is offering patients with advanced stage, non-curable cancers, palliative but yet very toxic therapies. is a service or a disservice in the current therapeutic landscape?
Um, so my father had extensive metastatic esophageal cancer, and he received chemotherapy, which made him sick. He died 2 months after the diagnosis, so it didn't really alter. In fact, it may have hastened his death, as far as I can tell. I think that's an individual. I mean, I don't know. I don't have an answer for that. I wish I had the wisdom to answer. I do not. One of the things I've learned from people that have pediatric sarcoma is that the parents often wish that they had not treated the kids because they suffer a lot and then they die anyway. But I think we all have to answer that question individually. What do you think?
Well, this is the question I'm faced with on a daily basis. Like
Yes, that's
the treatment, the disease that I'm treating, there's barely anyone who's ever cured, basically. So I agree with your assessment that this is, I mean, parents will tell us that even if there's 1 in a million chance, give it to my child.
sure.
But you'll be surprised how many 90-year-olds say the same thing.
Yes.
So life is a very precious thing.
Sure.
On the other hand, there are others who have total acceptance and are not willing to put up with pain for very little benefit, and they decide for themselves. So I agree with you that this is a very individual choice. But thank you so much. I'm very sorry to hear about your father.
That was, yes, a long time ago. And I think we've done a little bit better with esophagus, although I'm sure his fate would have not changed even in the modern era. But we're all touched with cancer. I mean, I think that's and it's a heartbreaking disease. I think, I mean, I'm happy to just make a contribution. If I can do that, I'll feel like I've done something. And certainly, it's something that these, as you well know, the patients really need to need help. And one of the things that I have the mathematicians do is make sure that they walk through the waiting rooms and see the patients because that's what we're here for. That's not a game. It can become a game when you're doing computer simulations. It's important to realize that when they talk about the burden of human cancer, it's substantial and it's individual as well as societal. And I think we need to appreciate that.
I'm so glad you added this part about walking through the waiting room. That's beautiful, and that's what we need to remind ourselves all the time.
Yeah.
Bob, it's been a wonderful experience listening to your excellent and thoughtful answers. And
Well, thank you. Thank you for doing this.
okay, thank you.